Okay, good morning, everybody. It's a pleasure to kick off the proceedings with the first panel today, where we're going to be focusing on cellular immunotherapy pioneers, liquid versus solid tumor strategies. It is so nice to be here in person, I have to say. I've missed seeing so many friends and colleagues from the sector, and I'm really looking forward to a great event over the next couple of days. And like Emil, myself, I also spent 6 hours trying to figure out what I was going to wear. But we made it. So without further ado, why don't I allow my fellow panelists to introduce themselves very briefly and their organizations. So from my furthest from me, Kanya, would you like to introduce yourself?
Yeah, no, thank you. My name is Kanya Rajangam. I'm the Chief Medical Officer at Nkarta Therapeutics, and thank you to the panelists and Oz for inviting me here.
Pascal?
Pascal Touchon, good morning everyone. Pleasure to be here, even though it's a bit strange to be like a fish in a fishbowl there. But pleasure to be with you. I'm the CEO of Atara Biotherapeutics, a leading company in the field of allergenic cell therapy.
And Devon?
Hi, good morning everybody. It's great to be back out in person again. And my name is Devon Shedlock, and I'm the Senior Vice President of preclinical or R&D at Poseida Therapeutics.
So with our panelists now, I'm going to go through in the next hour or so a couple of topics that we thought would be useful for the audience, as we're now actually, believe it or not, coming up to close to nearly a decade from when the first CAR-T therapy proof of principle data came out back in 2012. Wow. A decade's gone by, and within that time, remarkable progress, remarkable approvals, now seeing approvals globally, especially when you look at the Asian markets as well. But it'd be great to hear from you all individually, from a liquid tumor perspective, what's next on the horizon? Where are you focusing within your organizations? And what do you see as some of the key challenges for greater acceptance of the liquid tumor programs and platforms that are already out there? So Devon, why don't we start with you?
Yeah, I think that at least for the liquid tumor programs, that there are still some challenges that exist. I would say probably antigen escape. We see that certainly for BALL and also multiple myeloma. So I think some of the new strategies might be for dual targeting to mitigate or eliminate that ability to escape by the tumor. So I think that's probably one of the main challenges for liquid, if we're focusing on liquid.
Kanya?
Sorry,
Got a comment?
I can't actually hear too well with the plates here, but
so
no.
I'm going to use 3 microphones here because
Okay,
I've got 1, 2, 3.
you.
So if you give me your perspectives on what you see as the focus now for further acceptance for cellular immunotherapies within the liquid tumor space.
Great. So clearly the autologous CAR-T cells made a huge difference in the way we think of how we develop drugs and how we deliver it to patients. In terms of the main challenges there, one of it would be, I think, improving access across into the community, making them safer so you don't need specialized centers to be around to treat them. The other thing as well is the wait, because they're autologous therapies, so you've got to wait. And there are some estimates that only about 25% of patients who are eligible for these therapies are currently getting them. So a big part of the focus within the heme tumors, within the approved B-cell malignancies would be to make it safer, go out into the community, decrease the wait. We use NK cells as our cellular backbone. They have some inherent biologic attributes which I think make them well positioned to provide some of the answers for these questions. Beyond the liquid tumors too is then how do you identify other targets, say in solid tumors or even within the heme malignancies, something like AML, which is one of our programs. So I think those are some of the things that different companies have different approaches to try and make it be available more broadly.
Pascal, your thoughts?
Yeah, no, I agree with my 2 colleagues there, but clearly there have been a lot of progress recently on liquid tumors with the different autologous CAR-Ts that have been approved and used in patients there. I mean, my ex-colleagues at Novartis were mentioning 3,000 patients treated with Kymriah, which is great because many of them have been saved from what is a very sober prognosis there. On the other hand, there's still an issue in terms of accessing more patients and going in earlier line of therapies. And that's where we believe at Atara that allogenic cell therapy is really the way forward to be able to improve access and to go to earlier line of therapies at the same time. But there are also progress to be made in the level of CR, of complete remission, that is being obtained. Because if you look at the long-term CR, we're still stuck at around the 40-ish% there for all these CAR-Ts on CD19. Of course, it's higher on BCMA there, but certainly in lymphoma, it's still not at the level where we could say that we've done a job and that's fine. We need to go deeper than that. And there are ways to go deeper than that in terms of the type of cells, especially the phenotype of the cells, how we can make sure that we have more central memory phenotype that will allow to have different kinetics of the cells when infused into patients there. And also to be able to optimize the treatment, maybe in considering dual targeting. We're working on the CD19, CD20. We have an allogenic CD19 that's going to the clinic next year, CAR-T, that we really try to optimize for not only having rapid expansion, but the durability of the cells in patients and the ability to have enough persistence so we have durability of remission there. So we think there is a lot of space to grow in terms of the response, in terms of the level of response, the durability of response, and then in going allogenic to get better access for patients across the different line of treatment there. But it's great that we've made this first step with the first pioneer in the field. And here we are, I guess, in this room to work further and to expand that as much as we can, because we're not talking just about a few thousand patients. We're talking about 10, 20, 100,000 of patients that could be treated with these CAR-Ts and could be treated in earlier line. We've seen recently some data coming from autologous CAR-T going earlier and comparing with transplant. And clearly we have there the possibility to be much easier to use than a transplant in many ways, and having a high level of response and durable response. So a lot of work for all of us, but very excited to see that there is a way to get a best-in-class type of allogenic CAR-Ts and CAR-T in general to address liquid tumors. Now, we're also working on new targets because there are some new targets that are appearing that could be the answer to the antigenic heterogeneity there, and to make sure that we can address these patients that are not responding to like a CD19 CAR-T or maybe relapsing after CD19 CAR-T or BCMA CAR-T. So there are ways to work on new targets, and some new targets are starting to appear there. They are very exciting.
So you all touched on a component that actually one of the things that we discussed earlier is what we could be talking about in this panel, and that's around new target discovery and identification. And so, you know, I'd love to dig into that a little bit more with you guys as you think through this, because, you know, in the liquid tumor space, you know, we see, you know, the same targets emerging for validation for companies, the CD19s, the 2022s, BCMA. But, you know, the world is ready and pining for the next wave of targets to come through. So it'd be great to get a sense of how you are thinking about that. Are we really looking at, you know, improving and optimizing the current targets that we have with new, better tools and technologies, one approach, and combining with novel discoveries? And same for solid tumors, because let's face it, in terms of precedence and validation, there's a limit to what we have in the solid tumor space, and we'll come on to solid tumors in a bit more detail. But it'd be great to get an understanding of how you're approaching the problem of novel new target selection. So Devon, why don't we start with you?
Yeah, I think it's there's a lot in there to unpack, and the way Poseida is thinking about that, and I can't speak for all small companies, but as a small company, I think you know, we tend to be a little risk averse with new targets. Certainly, we and others like to go after clinically validated targets just to reduce or eliminate any surprises in the clinic. With that said, I do believe that CD19, 20, and 22 are solid and proven, clinically proven targets. And I do believe that there is quite a bit of room to continue to improve the tools and improve the platforms and the therapies to increase the durability of these drugs, and also persistence and engraftment. One of the things that we do at Poseida is we work with more of a prodrug T cell, and that is a very early memory T cell called a stem cell memory T cell. And we believe that that really could be the key to engraftment and durability, not only against liquid tumors, but also could be the key to potency in solid tumors as well. But these cells really are an ideal cell type for T cell therapies in that they're long-lived, they're self-renewing, and they're multipotent. So that's why we think of it as a prodrug, because once it's infused, it engrafts and then gives rise to your drug, which is your effector T cells, which then confront the tumor, and then once they're exhausted, there's wave after wave of effectors that continue until your tumor is gone. But back to target selection, I think certainly there have been a number of clean targets, including the ones that you have mentioned for liquid tumors. I think that the challenge will be greater for solid tumors. And maybe that is a great opportunity to utilize T cell receptors more due to the fact that a lot of the tumor antigens are intracellular. So there just might not be as many clean targets for solid tumor. One of the ways at Poseida that we're leveraging these types of approaches is considering our larger cargo capacity. So using the piggyback non-viral transposon system, we can encode multiple CARs. We've now shown proof of concept that we can co-express a CAR with a TCR. So by doing this, we think that we can not only reduce antigen escape, but also open the door to greater durability and efficacy in solid tumors.
And Pascal, I mean, Atara, established company, decades of research competencies and capabilities. How do you guys think about the framework for new target selection within Atara?
Yeah, I think similarly, when you are in a biotech company, you don't have the possibility to do too much translational work and discovery work there. So we rely a lot on academic teams or on other companies' work, both pharma and biotech, through orthogonal modalities. I mean, companies that work on antibodies, on CD3 engagers, on ADCs even there. And I think there is a kind of number of targets that we think is sufficient today for what we can do within our level of capacity and capabilities to address both liquid tumors and solid tumors there. So we're still working with some academic team about new targets. We have a collaboration with Michel Sadler at Memorial on some solid tumors targets that are at early stage there. We're working also with other academic teams about new targets in liquid tumors. But at this stage, we're really trying to progress our typical target like CD19 in liquid tumors, like mesothelin in solid tumors, to the point where we can go to the clinic and show the advantages of our platform in terms of leveraging the innate biology of EBV T cells coming from healthy donors, in terms of trafficking persistence of these cells, and then also working on the other aspect beyond the target, which is really the binder and the design of the CAR there, as well as the ability to address in solid tumors the tumor microenvironment and the immunosuppressive environment that the CAR are meeting there. Because it's one thing to get them to the tumors, traffic, and there, but they need to be able to expand and persist in the tumor despite the environment. And that's why we're working a lot on the design, like what we're doing for mesothelin CAR-T, where not only we have optimized the co-stimulatory domain with a domain called 1XX that is enabling the cells to be less exhausted, to be able to expand and eradicate tumors even after several antigen stimulation, several tumor re-challenge there. But then at the same time with a PD-1 dominant negative receptor, which is done not through gene editing of knocking down PD-1, but just adding that as part of the modification we're doing to the cells with the CAR to have that expressed at the surface, having a decoy for PD-1 that allows to have the binding with PD-L1 on the tumors, but at the same time no intracellular signaling, which is leading to some immunosuppressive signal for the T cells there. So all that is to optimize the condition into which we can express the activity on a particular target there. And we think there's still a lot of progress to be made there, because often there is an idea that, okay, if you don't see something happening like we saw with CD19 initially there, that means it's not working, the target is not adapted there. In reality, there is so much to do on the construct of the CAR-T, as well as on the ability to address the tumor microenvironment, that we believe that optimizing this is going to allow these targets that are well known, like mesothelin and others, to be able to then lead to very efficient and durable remission in patients with this type of lung cancer or mesothelin overexpressed cancer like lung, pancreatic, and ovarian cancer there. So a lot of work on that. But clearly at this stage, as a biotech company, we focus more on targets that have been already validated for different approaches and trying to optimize the way we address these targets.
Kanya and Nkarta.
Yeah. So I guess a couple of general comments I would say is when you're looking at a new or a novel target, when you go beyond the T cells, for instance, our cellular backbone is natural killer cells or NK cells, and our scientists would certainly argue that perhaps that allows us to leverage additional targets which may not be appropriate to put on a T cell. So it really starts with your cellular backbone, and to be sure that the target that you're choosing works with the cell that you have. And completely agree with what Pascal was saying in terms of optimizing. Once you have your target, your cell, then all the different components thereof in the CAR. The other thing I would add is that in Nkarta currently, what the approach we've taken is we have 2 co-lead programs. One is the well-validated CD19. The other one is unique and novel to us, and that's actually something that I'm quite excited about. Our lead program, in fact, 2 co-leads, but one slightly ahead in the clinic, has NKG2D as its target. Now, NKG2D is a unique NK cell target. There are companies looking at it at T cells as well, and they've got some results there, but we believe that it fits best on an NK cell because it's naturally found on an NK cell. It's one of the main activating receptors of NK cells. And what our scientists have done is taken that NKG2D and attached it to a co-stim and a signaling domain to really augment and increase the natural activity of an NK cell. That allows us to access other indications. Our first in human trial is in AML, notoriously hard to treat, both through cell or non-cell mechanisms, because it goes down the myeloid pathway. So this would be something which we are quite excited to see what it can do in terms of taking the promise of cell therapy beyond the B-cell lymphomas and so on. Going back to solid tumors, NKG2D as well has a lot of promise. The targets have been well documented across a variety of different malignancies, carcinomas, sarcomas, because they're essentially a marker of malignant transformation and very rapid division. Further, there's some work that's been published, most notably out of Baylor, which shows that when you look at solid tumors and the microenvironment there, the MDSCs, which potentially maybe are inhibitory, they are susceptible to the NKG2D signaling as well. So we are quite excited about our approach in terms of a novel target to try and expand beyond the B cell or the myeloma. Fingers crossed on what the clinical data shows. But so that's been our approach, is to do a kind of a co-lead program using our common platform.
Yeah. And I think we're very fortunate to have panelists today. The companies, the respective organizations are dealing with both liquid and solid tumor challenges. And so, past 5 years, I think we've really seen a confidence being established towards certain validated targets, be it PSMA or mesothelin, claudin, other targets, from a solid tumor perspective. And, one of the questions that I have is, can we actually cure solid tumors with T-cell-based products, be they CARs or TCRs? So I'd love to dig into that a bit more with you guys now. So Devon, why don't we start with you and your experiences from Poseida? Do you think we can get there with solid tumors and CARs and TCRs?
Yeah, I think that's the big question, right? I believe we can. I think that the proof of concept is out there. We've seen certain instances of that, of complete response against solid tumors. Now, I think one thing that was required to get there was we've seen these multi-dose regimens. But again, I do believe that T cells are endowed with the capability of eradicating a solid tumor. With that said, I think, as we continue to optimize our approaches and think about how to maximize that efficacy or potency within solid tumors, I think there's a number of approaches to that. I think a lot of folks are leaning toward the combinatorial approach with cell therapy, with IMiDs and other drugs. As I was mentioning before, at least at Poseida, we're thinking about this stem cell memory CAR T, which is really more of a prodrug, and that sort of has waves of effector T cells that are packed into a single dose, but essentially achieving that the way we think about it is achieving the same thing as a multi-dose regimen. But I think that there is a lot of potential there for treating solid tumors, and I think that we'll ultimately be able to get there. Now, our experience at Poseida is fairly limited. We do have a program targeting PSMA and prostate cancer, and we're about to file on a MUC1C targeted CAR T. But with this limited experience, we've seen very promising data within prostate cancer. And we've seen so far very few patients at our first 2 doses of the trial, but we've seen several instances of over PSMA 50 or PSA 50 responses. And we're very excited to see that even one of our patients was able to achieve almost a complete response. And mind you, that I believe was with a CAR T dose of 22 million CAR T cells. So extremely low doses, but they really were able to pack a punch. So I think as we continue to progress and improve the technology, I think that we'll see that this will become more and more achievable. And as mentioned by my colleague, I think also moving earlier in the line of treatment will also increase those response rates that we've been seeing currently. So
Thanks,
that's
Devon.
really our thoughts.
So Pascal, can we cure with TCRs and CARs, and what do we have to do that's different versus liquid tumors to achieve that goal?
I'm very optimistic, and I'm very optimistic because of immunotherapy in general. I mean, we've seen with checkpoint blockade that at least some patients, between 20 to 40%, depending on the disease, are able to mobilize their T cells and control the tumor there. That's what checkpoint blockers are doing, basically. And here we have the possibility to bring some new T cell to these patients there. Not only to the patient that have been responding, but the patient that have not been responding to checkpoint blockade there. And I think that is fundamentally why I'm optimistic, because it's really addressing the fact that one of the reasons they might not have been responding to checkpoint blockade is because they don't have enough T cells, not enough activity T cells, or there is a need to boost their system into bringing these T cells there. And especially with allogeneic T cells, whether it's allogeneic TCR-T or CAR T is another debate there, we're really bringing that boost to the patients. And having these T cells, especially when you optimize the construct in a way that address the tumor suppressive environment there, you then have the possibility to get the expansion and eradication of the cells. I mean, we've done some preclinical data on mesothelin CAR T, allogeneic CAR T there, which is quite remarkable in saying that just having changed the design in terms of the binder, the co-stimulatory domain, and the PD-1 dominant negative receptor, allows the cells to expand and eradicate tumors even up to 10 and more tumor re-challenge, whereas a traditional CAR T will be exhausted after 2 or 3 tumor re-challenge there. So there is a way to optimize, there are ways to address access through allogenic CAR Ts there, or TCR-Ts, there are ways to optimize the natural biology of EBVD cellular technology platform there. All of these are ways to address the tumor microenvironment and the barrier put to the T cell activities there. Now, one of the questions, of course, that needs to be also addressed is whether these type of T cell that you bring, especially allogenic one, could you redose? And we've seen that is possible, you can redose. And therefore, how do they integrate themselves in a treatment paradigm? And do you combine them with other type of immunotherapies? Do you sequence them in a particular way there? So there's still a lot of work to do, and that's why we're here, is to discover new ways to use these T cells there. But clearly, mobilizing the T cells with checkpoint blockade has worked, bringing new T cells to these patients in a way that allows the proper level of trafficking, expansion, and persistent, we believe is going to work. And then we were going to optimize the way to address that in terms of the treatment regimen and paradigm in combination and sequencing in the right way with other approaches. So I'm extremely optimistic that we'll get there.
Kanya?
Yeah, so in terms of solid tumors, some of the challenges have been the target, then the tumor microenvironment, and I know we are bucketing solid tumors as one, but obviously there's variability in terms of how permeable they are, and even across patients of the same tumor type. And another part as well has been the trafficking to the tumor site. So the way we are thinking about it for our program is for our solid tumor with NKX101, that's the NKG2D that I just talked about, is we hope to start by giving it locoregionally through the hepatic artery for liver localized disease. There have been some small works that have been done showing radiotagged cells when they're given through the hepatic artery preferentially localizing in the tumor rather than the surrounding liver, because the liver has a dual blood supply and the hepatic artery preferentially vascularizes the tumor, the portal vein, the rest of the liver. So leveraging some of those, that's a standard of care technique. People are using it now to give radiation and chemotherapy. So now, why not cell therapy through there? That's one. But when it comes down to, being broadly looking at solid tumors, it comes down not only to the targets, but the combination. And going up front, as I agree completely with Pascal, whether it's solid or liquid, ultimately it's about combination therapies to increase the likelihood that a patient would respond, achieve a deep response, and be durable. I think the other point Devon made earlier on as well about multiple dosing. I'm biased, obviously, but NK cells with their safety profile allows you to do that. So these cells inherently don't explosively divide when they see a target. So the expectation is things like CRS and ICANS and the cytokine outpouring is going to be more modest. So which means that you can do multiple dosing. In fact, both of our trials, we give 2 and 3 doses after one LD chemo. So that's some of the advantages as well as you go beyond the T cell paradigm for cell therapy.
So let's dig in more into sort of the allogeneic space. So our colleagues at Allergene last week very proactively, very quickly described an unfortunate event, which actually in the field of cell and gene therapies is not unusual to get the kind of chromosomal inversion that they saw, and they will in due course, of course, get out with the full narrative and the details. But as you think about now the allogeneic space itself, when do you think we're going to see the first approvals coming through for liquid tumors, for sure? So Devon, what's your prediction there?
Yeah, that's a great question. I think first and foremost, we need to see safety of these therapies, the allogeneic therapies. I think safety is paramount, but then I think the level of efficacy, I was speaking to someone the other day and they said that they want to see at least 75% the level of efficacy with an allogeneic cell therapy that you would see with auto. So unless it's 75% or better, that's what they think that, where sort of the cutoff line is for an allogeneic therapy to be commercially viable, which would in that case then cannibalize the auto therapies. But safety, again, I think will be paramount. And it's unfortunate about the event at Allergene, and I think we still need to learn a lot more about what has happened in that specific case. But I think a number of factors can possibly contribute to some of these safety events, considering things like virus to create the product. Another thing that we were possibly hypothesizing about was that if the cells are stimulated before they're edited. So for example, if the cells are activated, stimulated, and start dividing before they're either transduced with a virus or gene edited with a gene editing technology, there's the possibility that cell division can exacerbate chromosomal instability when cytokinesis occurs before double-strand breaks are fully repaired, which could lead to that aberrant chromosomal segregation. So at Poseida, this is something that we've been thinking about for a long time, considering our fully non-viral platform, but also the fact that both our piggyback integration technology as well as our Cas-Clover gene editing technology work completely in fully resting cells. So we do all of our integration and gene editing before the cells are stimulated to divide. So again, I think we all have to keep our eye on safety, but you need to see that these products are safe and they have to be effective.
Pascal, timings, you think, for hematological approvals, at least with Allergenics? That's a subject close to your heart, I know.
Yeah, I'm biased because, of course, I believe that the first approval will be next year of Tab-cel, an Atara product that is going to be filed in Europe in November. And that's the first allogeneic T-cell therapy that is going to regulatory bodies there. And we plan then to also file for BLA next year there. So we think that this could be very well the first allogeneic T-cell therapy to be approved. It's being developed for the phase 3 into a very specific ultra-rare condition, but very serious condition called PTLD, which is a post-transplant type of lymphoma that is EBV-related, Epstein-Barr virus-related there. And we've shown in previous studies in phase 2 very high level of response and durability of remission, having usually 50% of the patients being treated responding and having the ability to have an overall survival that is around the over 80% or 86% and over after 2 years there, where these patients are usually of life expectancy only 2 to 3 months as a median overall survival there. So it's truly a transformative therapy, and we're very exciting to file very soon now. Now, to address the question of the field in general, and because that's not an allogeneic CAR T, it's an allogeneic T-cell therapy, but not an allogeneic CAR T. I mean, it's true that the unfortunate event, I mean, we know how professional is Allergenic Therapeutics in the way they've dealt with that and the way they are trying to understand what's happening there, is raising some question. But that's the case with many types of transformational therapies when you do these new technologies. Any case is creating some question from a safety point of view there. The key question, and we know that the team is working hard to answer this question, is whether this particular issue, which is relatively rare, 1 out of 100 patients, if we understand well what the company has said, whether it happened before, during manufacturing, or after manufacturing. And I think that's a key aspect that the company is having to investigate right now with the FDA, working, I guess, in collaboration with the FDA. And that's going to have also different impact on the field for allogeneic CAR Ts there in terms of what type of control, manufacturing control and release criteria you need to have to be able to look at that type of situations there. Now, as my colleague was saying, Devon, I mean, there are different types of technologies. So it's difficult today to say that this is a potential issue for the field. I don't believe it is. I think it's something to look at, of course. But there are very different types of technologies. For example, at Atara, we don't gene edit the TCR because we leverage the endogenous, native TCR from the EBV T cells, and we just add a CAR. So there's different level of modification of the cells that might lead to different type of variability of what you get and different type of risk and safety aspect that you might envisage there. So I don't believe myself is a systemic issue for allogeneic CAR T. I think it's specific issues, but of course, key answer will be when the company knows and communicate whether this has happened during the manufacturing or post-manufacturing in the patient, because we are developing, whether it's autologous or allogeneic in cell therapy in general, living drugs. I mean, they have their own behavior when they are infused to patients. They expand, they disseminate themselves, they traffic, they have their own ability to develop an activity, be it on efficacy, on safety. And we've seen how different, for example, the safety profile of allogeneic CD19 CAR T is compared with autologous CAR T. And that's explained that there are huge differences between these different products that have an impact on safety there. So we hope that that will be resolved soon, and we wish the best, of course, to Allergenic Therapeutics there. And we're looking forward for a clearer answer to that question. But I'm not worried about the field in general because, again, very rare event, to be confirmed and controlled, and then we have answers for different type of technologies to address any type of issue that might be encountered there.
Fully agree. Kanya?
Yeah, so maybe just to continue on the question of the Allergene Therapeutics and the recent clinical hold last week, you know, based on what the company has said so far, it does appear to be just clinically, perhaps, you know, they saw some cells that were that had a clonal abnormality and some outgrowth and pancytopenia. So almost, I hate to use the word incidental, but a finding on a biopsy. But what was, it's early days, obviously, and you know, I have no doubt the smart folks at Allergene will figure it out. But one of the questions that I have in my mind is, the hold was applied across the pipeline. So I wonder how much of it is the FDA looking for, is there a release assay which could have or should have picked it up? And I think that's the bigger question for the field, because how do you come up with an assay which is going to pick up a chromosomal translocation that manifests 6 months later, right? So that is something that requires a little bit more thinking. In terms of the gene editing itself, NK cells, one of their big biologic advantages is they don't express a TCR, so they lend themselves to being allogeneic because you're not worried about graft versus host disease. So, but having said, so neither of our first 2 programs in the clinic have, I'm sorry, someone's calling me. So neither of our first 2 programs in the clinic have an edit, but we want to include edits. In fact, our 3rd program, we are planning on a couple of them. So this is something that as a field, we all have to figure out, especially if what the agency is looking for is some sort of an assay, and maybe Devon can weigh in here about how, would there be an assay like this which can predict, because translocations and so on do happen when you use these techniques. So how do you figure out what's going to end up causing an issue like this, and what perhaps is just, you know, not clinically insignificant.
Yeah, and do the translocations actually have any clinical consequences at all? I mean, that's recognized with TALENs, with CRISPR. I mean, you know, we published at Tmunity with Carl at Penn, you know, that you see these translocations. The question is of clinical significance.
Right.
And knowing the team at Allergene and how proactive and smart that team is, I think, you know, we wish them well for the sake of their patients to get the clinical hold lifted as soon as possible so we can get back to the business of treating patients. You know, one area that we can't avoid talking about as we look at cellular therapies, be they autologous, allergenic, liquid to solid, is the thinking around manufacturing. So as you now, you know, clearly, Pascal, your organization, very established, big footprint in manufacturing. Devon, also, you know, you're building out in that regard. Kanya, you guys, of course, thinking about scale for your NK platforms. What's constant in terms of the CMC thinking, whether you're dealing with liquid or solid tumors? And then, you know, the trickier question, are you guys thinking of coexistence, or are you thinking cannibalization when it comes to allergenic and autologous products? So two-part question there. The first of all, what's the same when you're thinking about manufacturing scale, CMC know-how, quality matters, et cetera, be they allergenic, autologous, liquid, solid? And then secondly, can they coexist or not? Devon, let's start with you.
Okay, liquid solid. I don't think there's really much difference from a manufacturing point of view if the difference in your product is just binder. So if your target is different, then I don't really think there's much difference. But I think if you are incorporating new technology or additional technologies in your product in order to enhance its potency or, for example, counteract the immunosuppressive environment within the tumor microenvironment, then I think that could increase the complexity of manufacturing. Auto versus allo, I think obviously, you know, most of us have all felt the pain of creating a personalized medicine on a patient-by-patient basis. There are many hurdles and challenges to doing that, and so there are great efficiencies in manufacturing the allo product. At the end of the day, I mean, allo has to be safe and it has to work. But with that said, I think the most exciting aspect of that allo manufacturing is the ability to achieve multiple doses from a single manufacturing run, and that could be in the tens, that can be even up into the hundreds. That's probably what is very exciting for us. And then I think further down the road, especially with allogeneic manufacturing, depending on your cell source. I mean, obviously, if you're starting from a single cell, if you starting from an iPSC-derived type product, your manufacturing is probably a bit easier. But if you're starting still from a raw material that's being derived from a healthy donor, I think we start thinking about automation, and we start thinking about ways to reduce the number of touches, completely closed systems, because time really is no longer a factor. I mean, you don't have to really worry or factor in that vein-to-vein time any longer. So you create the product, it goes on the shelf, and it can be used then in any patient at any time. I think that's really the most exciting part about that. But you can do your due diligence on that product to look at all the safety questions and perform all of the release assays that Kenya was describing to make sure that they are safe but also effective. And I think the field is really, we're all wondering, how do you assess that? How do you assess or evaluate how a product is going to perform in the clinic? And I think that's something that, obviously the FDA doesn't guide on, because they're more focused on safety. But how do we actually assess that? And, we find at Poseida that really it's almost no in vitro assay is going to predict how that product is going to perform in the clinic. And so we rely heavily on in vivo models, which are just complex systems, and we don't necessarily believe they're going to predict performance in the clinic, but it's going to tell you about how robust your product is. Is it, what is the fitness? How healthy is that product? What is its capability of expanding and so forth? So I think these are all the things that we have to think about when manufacturing both auto, but now moving into allo.
And Kanya, clearly you're in the allogeneic space, but love to get your perspectives as you think about scale for the platforms, and then thinking, is there going to be a coexistence, or is it going to be ultimate allogeneic? Well, I'd love to get your perspectives on that.
Yeah, so I think one of the key differences with allogeneic versus autologous is going to be cost. I mean, our manufacturing folks are saying cost of manufacturing for a patient would be about $2,000, which is just unimaginable when you think of autologous, because you can scale up instead of scaling out. So if you're thinking of autologous and 20 patients, then you need 20 cell cultures, as opposed to just scaling out and making more doses from one lot. So I think that's a big advantage as well. The field is evolving towards allogeneic, different approaches to get there. One of the advantages perhaps of autologous is that it's one and done. So you give one dose, it's something that's intrinsic to the patient, so you don't have to worry about host rejecting the cells and so on. So perhaps there's a niche there way down the road to continue to use it in some selected patients. But with all of the exciting new technology that folks in this room are working on, different cells, different targets, different ways of putting in the constructs, I do believe that the field is going to end up being allogeneic almost all the time, and fairly quickly. As Pascal was mentioning, Atara has got the first allo product going up for review next year. So it's happening now, and it's only going to accelerate.
So Pascal, you have a very unique position and experience, right? You and I both, with the first autologous CAR-T therapies, right? The trials and tribulations there from CMC manufacturing, and now thinking about your world of allogeneics. Give us your perspectives on what do you think is similar and what is really going to change, and will one trump the other at the end of the day?
Yeah, I think, first of all, autologous CAR-T are there. They are proof they are saving lives. So yeah. I think that's a fact. Okay. Yeah. We are working all on allogeneic that are not yet approved, and we hope to be approved and to save lives we believe, in a more convenient way on the existing targets there. I think it's been mentioned by my colleagues, one of the key aspects is access for patients, is the ability to make that at scale. And that depends, there is no difference between liquid and solid from that point of view. It's when you start with material, whether it's donor-derived or iPSC or whatever, there you can make product depending on the target, depending on the construct, and depending on what type of profile you want on the ultimate product in terms of phenotype of the cells. Do you want more central memory? Do you want more effector cells? There are a lot of variables, but it's not really linked with solid or liquid tumors there. What is important in the scale is to have the technology that allows you to produce that at scale. And for example, at Atara, we've developed now manufacturing into bioreactor of the cells. And we have done that at a level of up to 10 meters scale bioreactor now. And we can produce a lot of cells of good quality there. I mean, in fact, we believe for our multiple sclerosis product, because we are not only in oncology, we are also in autoimmune disease, and we present tomorrow some very exciting data on 8188, a product in multiple sclerosis in patients there. And for that product, we know that ultimately we'll be able to make 20,000 doses from one leukopak. So that's a type of scale up that we can make with this technology of bioreactors that will bring, in fact, cell therapy very close to monoclonal antibodies in terms of the efficiency of manufacturing and therefore the cost of goods manufactured, which I think will be a great progress there from that point of view. Now, coming back to the autologous versus allergenic, I believe in both, but I strongly believe that on the current way we're working, especially at Atara, we're going to displace the autologous CD19 CAR-T or other type of CAR-T there. Now, there are some aspects where autologous might be quite helpful, especially when you have more of a multi-target type of approach there. Because in the allergenic field, you need to make one particular target, one particular product there. So if you need to have a mix of different cells in terms of addressing a more polyclonal type of approach to targets there, that way autologous might still have a place, I believe. But ultimately, if you have only one particular target, we believe, and we have to prove it, so don't get me wrong, we are not yet there, but we believe that the allergenic CAR-T will really take the place of the autologous CAR-T if we are able to show safety and efficacy at least at the same level, if not better. So that's the belief, not yet evidence.
Thank you very much. So that concludes the main session of this introductory panel. I think we're running on time. Do we have time for one or two questions? So I'll open up the floor if there are any burning questions from the audience to my fellow panelists. If there is a microphone or somebody has a question, I'm blinded by the light, so I can't see very well here right now.
All right.
Any questions for the panel?
I have a quick question around the allogeneic versus autologous. As you scale doses from 1s to 10s to 100s, how do you think about the formulation filling challenges of being able to cryopreserve bags of 100 doses out of a batch and keeping the product quality during that process? Seems to be a challenge that there's not a lot of good solutions to manage today. Thank you.
So if I can just paraphrase the question, if I heard it correctly, this is regards to sourcing cryopreservation for large batches for allo. That was the question, correct? Yeah. Any comments, any perspectives? Do you see it as a challenge?
So maybe I'll start there.
Yeah.
So I think if I understood the question, it was about, when you scale it, you're scaling up on these huge volumes, how are you going to source all the materials? Is that managing the process? Yeah. Yeah. No, thank you. That's helpful. That's actually interesting. So I'm not the manufacturing person, but my manufacturing colleague very early on made the decision to go to vials for, I believe, that reason, which is that when you are thinking of scaling out and doing all of these things, then it's not sustainable to put it in bags. So I think in that seems almost maybe too easy an answer, but that's what we did. Is we very thoughtfully, so early on we had some good discussions internally about, hey, all the clinical sites are used to seeing these cell therapies in bags, and he was pretty firm about, hey, if we've got to do thousands and thousands of doses, vials are the way to go. So that's what we do, and that works for us so far.
One more question. Time for a question? Everybody's ready for coffee, obviously. Okay, well, Kanya, Pascal, Devon, thank you so much for engaging today on this great panel. Please thank my panelists.